everybody, it's Professor Alfred here, and if you're getting this video, it means we have another virtual lab, and we are going to be finishing up with Chapter 13, which is the anatomy of the nervous system. I'm going to get into neuroanatomy here, specifically the central nervous system this week. So let's go ahead and get into it. I always give a why it matters slide. So Parkinson's and other neurodegenerative diseases. When I mean neurodegenerative, any type of pathologies that affect neurons and nerve cells and lead to certain abnormalities. And remember, structure equals function, so an abnormal nerve cell could affect the function of the brain. So for Parkinson's disease, we know it as a neurodegenerative disease that causes uncontrollable movements such as stiffness, shaking, difficulty with balance, and coordination. Some signs and symptoms that we see with these patients can occur gradually and worsen with age over time. And it can lead to difficulty walking, talking, and it could even be behavioral problems such as sleep problems, depression, memory, difficulty remembering things, as well as fatigue. And the demographics, usually it affects older population, so usually after age 60. An early onset for Parkinson's diagnosis could be hereditary related. And we've also seen in cases that it affects men more than it does women. So the etiology, although unknown of a source of why this happens, we know that when the nerve cells in the basal ganglia, which is pictured right here, draw my little pointer, this is your basal ganglia, any type of degeneration of that, pretty much when it becomes impaired or dies, those cells, that's what affects this, because the basal ganglia is responsible for movement and produces dopamine. So we said that one of the symptoms is uncontrollable movements, so damage to the basal ganglia could cause that. And then what causes the neuron to die, again, is still unknown, but overall the combination of genetic and environmental factors are linked to a causation for this pathology. All right. And so why does that matter? Again, we need to learn about neuroanatomy so that we can properly diagnose our patients, understand how things occur, and the structure and overall function of the body. So these are your learning objectives that, pulled right out of your textbook. My biggest advice whenever you're trying to study, not just for lab, but lecture as well. If you can answer these enabling objectives, you're pretty solid on this. So we're going to get into these, but basically by the end of this lab, you'll be able to tackle some of these on an application basis. All right. So recap from last week. I know we kind of did a little lecture on this, but just to go into it, the central nervous system versus the peripheral nervous system, you need to know the distinctions. Remember, the central nervous system is the brain and spinal cord, and the peripheral nervous system is everything else. We're going to focus on the CNS this week. And remember, a collection of cell bodies for the CNS is the nucleus. And then we've got your gray matter and your white matter. And to get into that, remember that our gray matter is unmyelinated, and it's kind of used for information processing. And for the brain specifically, for the central nervous system, that's the outer cortex of the brain. So right here, see the gray matter is the outer ridges, whereas the white matter is on the inside of the brain, okay? So gray matter is information processing. It's unmyelinated. When I say myelinated, that's your white matter because it's a lipid fatty layer for communication. So it's for cell signaling for the neurons. So if you can see, this is actually flipped for the spinal cord. All of this is from your textbook, by the way. This right here is white matter. Now it has a special stain to it, the histology, the histological slide. But even though this is darker looking, this is actually your white matter. And then your gray matter, which I'll do in green here, is this butterfly looking aspect of your spinal cord, which we're going to get into the name of that, your horns of the spinal cord. But yeah, it's important to know the distinction, how, like I said last week, this is what the spinal cord looks like, gray versus white, and then this is what the brain looks like, gray and white. Another recap from last week. Again, I'm kind of speeding through this because we already went through it, but it's very important that you know the organelles and the parts of a nerve cell. Specifically, we're talking about cell signaling. We talk about the axon. So remember, the neuron is for cell-to-cell communication, and your dendrites right here, see those little hair-like projections, will collect electrical signals from the synapse of another neuron, which you see right here. And then those signals are sent to your soma, which is a cell body. And that's kind of where the information is processed and determine if the neuron's going to fire the action potential. And that goes through your axon right here. And that cell signaling is further sped up, I guess, or motivated more efficiently because of your myelin sheath and your nodes of Ranvier or nodes of Ranvier. I've heard it two different ways. It's a French name. But Remember that your nodes around VA are the gaps in the myelin sheath. So these are here. Boop. And those kind of help through saltatory conduction speed up rapid transmission of that electrical signal. And your myelin sheath is an insulation for your nerve cells to also allow for that rapid transmission. And then that's your synapse right there. So it's important you know those parts. So axons, the nerve versus the tract. It's very important you know the difference. I'm using the eye, this slide that's on your textbook as well, as a good example of it. But an axon, like we said, transmits that electrical impulse from the neuron cell body, the soma, to other cells, right? So if it's a bundle in the peripheral nervous system, that's called a nerve. So an example of that are axons that leave the eyes, that bundle of axons that leave the eye, that's called the optic nerve. So that's leaving. But if there's a signal going towards the brain, so that's called a tract. And an example of that would be the axons from the eye entering the cranium, which is your optic tract. So your tract goes this way, and then your nerve goes that way. It's kind of important you know the distinction with that, because remember, terminology matters here, okay, guys? Then we've got the meninges. Now I'm introducing some new stuff. The meninges is kind of like remember, connective tissue is for support. This is a membranous connective tissue. Here's the image right here. And it's the outer surface of the brain, and it's composed of 3 layers. Best way to remember this, and this is how I remembered it when I took anatomy, you've got your dura mater, which is Latin for tough mother. That's the outer layer. It has to be tough for a reason. It's a thick, fibrous protective sheath, and it pretty much encloses your central nervous system and all major vasculature of the cranial and vertebral cavity. So all those extra blood vessels underneath and stuff, your dura mater is like kind of closing that, right? That's the outer layer. Then in between is your arachnoid matter. When I think arachnoid, think arachnid, as in the thing I'm afraid of. All jokes, but the arachnoid matter, it's got its name for the spider-like, web-like appearance of it, because it kind of has an interlocking between the dura mater and the arachnoid matter is the subarachnoid space, which contains, it's right here. And you see that right there? See how spidery it looks? But yeah. The subarachnoid space contains cerebral spinal fluid, and that's important for cushion protection, especially for your brain. And we'll get into like an importance of that like in the clinic later in a second. But important to know that subarachnoid space, your arachnoid matter right here, that kind of has that spider-like projection. It has your CSF. And then under that is your pia mater, which is very thin, very gentle. How I remember the pia mater is it's Latin for tender mother. So you've got your dura mater, which is the tough mother outside, and then you've got your pia mater on the inside, which is your tender mother. And it's like a thin fibrous membrane, and it covers your gyri and sulci. Your gyri, your ridges, and your sulci are your grooves. That's the stuff right here. So it pretty much covers that part of the brain, of the cortex, okay? And there's some important parts of the meningeal layer. Now, for basics on neuroanatomy, I'm not going to go deep into it because I know you have it for lecture, but the cerebrum and your cerebral cortex is composed of like your gray matter. And a big thing, taking it back to chapter 12 a little bit, when it comes to embryonic development, the telencephalon, it expands and becomes your cerebrum through that development. Embryonic development means from gestation to an adult, like, certain parts of our body grow and specialize. That's part of it. So there's 4 main lobes demarcated by your large gyri and sulci. What's important to know is that there's no distinct border between these 4. So my camera is kind of covering it, sorry, but this is figure 13.6 in your lab. Perfect image to study from. You guys have my slides for the 4 main lobes of the cerebrum. And it's the frontal, parietal, occipital, and temporal. And the importance of me saying that there's no distinct border between them, again, structure means equals function. If there's no distinct demarcation between these lobes, it means they communicate with each other and their functions are interrelated. So what I mean by that is like the frontal lobe also uses some stuff with the parietal lobe kind of thing. It's not just like only the frontal lobe is working at one point in time. It's all commingling together and communications are going together, which we'll get more into that for part 2. But the importance is that, structure equals function, okay? So the frontal lobe is for your complex executive functions, and your prefrontal cortex is for decision making. So way to remember this, recent clinical studies for like behavioral changes or psychopathic patients, for example, have impairment to their prefrontal cortex, their decision making. So that's a good way to kind of remember that. Again, structure equals function. If I get into a major motor vehicle crash or trauma crash or some type of traumatic event and I damage my frontal cortex or my prefrontal cortex specifically, that will affect my decision making. And then you got your parietal lobe, which is for somatosensation. That's where it's processed. And then for occipital, a way I remember this, let's say you fall on your back and you fall really hard and hit your head. The first thing you do, they always say in like, what is it, in cartoons, when they fall and hit their head, they see stars, like the little cartoony stars. That's because it affects your vision when you hit your head that hard in the back, because the occipital lobe is for vision processing. And then your temporal lobe, how I remember this because it's so close to it, your temporal lobe is for auditory processing and memory. And then there's this lobe that's kind of like deep. It's the fifth lobe of your neuroanatomy for your cerebrum. Yes, there's 4 main lobes, but there is a 5th one called your insular lobe. Not really well understood, recent studies on it, but it is linked to processes such as your motor control, homeostasis, which we know that means like the balance of your body to stay alive, very important, and overall self-awareness. So it could be linked to addiction and neuropsychiatric disorders as well, we've seen. Again, not well known, but it is a 5th lobe that's important to note because it's in your textbook. Now, the diencephalon is a connection between your cerebrum and your nervous system. There are 2 major regions that are highlighted right here. You've got your hypothalamus and your thalamus. So your thalamus is a collection of nuclei. Remember, nuclei are cell bodies in the central nervous system. Remember last week? So it's a collection of nuclei that kind of relay information between your cortex and your peripheral nervous system, as well as spinal cord and brainstem. All sensory information except olfaction, which is the sense of smell. So every other sensory information passes and is processed through your thalamus, with the exception of the sense of smell. Very important I say that 2-3 times because it's definitely a high yield information to know. Now your hypothalamus is a collection of nuclei that are responsible for homeostatic mechanisms. So it's in charge of the autonomic nervous system and the endocrine system. So that's where we get into homeostasis, because your endocrine system, it's kind of in charge of that via your anterior pituitary gland. We'll get into hormones and stuff and homeostatic regulation in part 2. But that, like I said, that's where it's kind of localized. And then also linked to memory and emotion, kind of like that processing via the limbic system. So that's the importance of these 2 components of your brain. And now let's get into what we call the lizard brain. We call it the lizard brain because it's the most primitive form of your brain. And therefore, basically, major functions are kind of to maintain that you're alive. They're not so much for higher order thinking like your cerebrum is and your frontal cortex. This is more like main stuff to keep you alive, which is why we call it the lizard brain, like nitty gritty. We already went over the basal ganglia, but 2 other components of it are your brainstem and your cerebellum. So your brainstem contains the midbrain and the hindbrain, your pons and your medulla oblongata, right? So the midbrain is visual, auditory, somatosensory perception. That's kind of where it's located. The upper portion of your midbrain is the colliculi. That's Latin for little hill, which kind of describes what it looks like because it's kind of called little hill for the 4 bumps that it has. And the inferior colliculus is the auditory pathway. And the superior one is a combined sensory info for visual, auditory, and somatosensory. And then I just misspelled it, my bad. And then you've got the pons and the medulla. So again, that's for cardiovascular, respiratory systems and rates. Your medulla oblongata, part of your brainstem and stuff. Think of it keeps you alive, like breathing and stuff. And then your cranial nerves kind of connect through the brainstem. Continue on the lizard brain. Now we're going into the cerebellum, which is your little brain, literally Latin for little brain. And because it looks like a little brain, it's located right underneath your cerebral cortex. It looks like this. And it has the gyri and sulci, just like your cerebral cortex. And it kind of compares the information from your cerebrum to the sensory feedback from your peripheral nervous system through the spinal cord. And then you've got this inferior olive that is for fine motor coordination. All right, cranial nerves. I'm not going to go deep into this, but you do need to know this for lecture This is a beautiful table that is in your lab that I definitely recommend you kind of at least glance at. And they're located in the brain in certain parts for a reason. A great way I recommend memorizing your cranial nerves is recognizing also kind of what lobes or part of the brain they're in, because it also helps you remember what part of that brain, like what it specializes for, you know? And so I also have a mnemonic for you. This is what I learned from like, you know, online and stuff, me trying to study. You've got to have to remember some of these things. If you do have to memorize the nerves, the cranial nerves 1 through 12, mnemonic I learned is on. They traveled and found Voldemort guarding very ancient Horcruxes. Yeah, if you're a Harry Potter fan. So the first three on is olfactory, optic, oculomotor, and then they traveled is trochlear and trigeminal nerves. And then and found is your abducens and your facial. And then Voldemort guarding is vestibulocochlear and glossopharyngeal. And then very ancient Horcruxes is vagus. That's a big one, cranial nerve 10, by the way. You're going to get some questions on that. Accessory and hypoglossal, okay? I think the biggest ones that are brought up a lot, vagus nerve, especially when we get into like other mechanisms as well for the nervous system. Now, anatomy of the spinal cord. I'm going to brush over this too. Name coincides with the vertebral region. So, again, kind of like I said, with like intercostal muscles and then your intercostal space and your rib cage, like those names are kind of together to show that they're kind of localized together. Same for your vertebral column. So you've got your spinal cord. The region you have cervical, thoracic, lumbar, and sacral, just like your vertebral column. C1 through C7 of your spinal cord are above the cervical vertebral named counterpart. So what that means is C1 is above your C1 vertebrae, and then C2 is above your C2 vertebrae. And then it keeps doing that all the way until it gets to C8, and that's what changes it. Because at around C8, which is right here, that's between C7, so it's below C7, but it's above T1. And because of that, after that, from T1, your thoracic spinal cord, all the way down, every single name after that is below its vertebral counterpart. So what I mean by that is C8 is between C7 and T1, and then T1 is below T1, and then T2 is below T2, and then T3. That's how you can remember it, so you know the location. From C1 to C7, it's above its vertebrae, and then C8 makes that change, and then you've got T1, which is below T1, all the way down, okay? So that's a big trick I learned when I was taking A&P. So we also have major enlargements, your cervical enlargement, as well as your lumbar enlargement. This has to do with our plexuses. Your C3 to T2 is your cervical enlargement, and that's because of your cervical and brachial plexes. And then your lumbar enlargement from T7 to like T11-ish has to do with your lumbar and sacral plexes. And then your conus medullaris, because we have to have something for the sacral region. Right down here in the sacral region is your conus medullaris. That is where the spinal cord kind of tapers. When I say tapers, it's going as this tube this whole time, right? And then around the sacral region, it starts to kind of taper off like a pencil, basically a little, comes to a point. And that's the conus medullaris region. And then right after that, there's this bundle of nerves that are long and projected right after the conus medullaris. And that's called the cauda equina. It looks like a tail, so it's called cauda equina because it is Latin for horsetail, because it looks like a horsetail. But it's pretty much responsible for the movement and sensation of your lower body. And then you've got the filum terminale, which kind of keeps the cauda equina, which is that little horsetail, attached to your coccyx, you know? So it kind of anchors it to the coccyx. So it's very important that that's there. Now, for the meninges in the spinal cord, remember I was going to bring it back up, the cerebral spinal fluid? This is where it matters in the clinic. So for your subarachnoid space, remember I said that it has cerebral spinal fluid in it? If there's an inflammation of that, or if we're trying to rule out meningitis, we'll perform what's called a lumbar puncture, and that's the removal of cerebral spinal fluid to test for inflammation and infection of the meninges. And it kind of happens right here. your lumbar region, obviously, lumbar puncture, and they take a long needle and kind of pull out some cerebrospinal fluid. If there's some type of bacterial growth or inflammation or infection, that could be positive for meningitis. Now, meningitis is inflammation of the meningeal layer due to some type of viral or bacterial infection. And some signs and symptoms are fever, chills, nausea, vomiting, sensitivity to light. You're like, okay, that could be anything. But a big one that's like a pearl for it is nuchal rigidity or knuckle rigidity. But that means nuchal means neck and rigidity stiffness. So if someone's got fever, chills, nausea, and all that, and their neck is very stiff, we start questioning, oh, do they have meningitis? And then also an altered mental status could lead to that too. Yeah. Cross section of the spinal cord. So here is a cross section right here. And we've got gray matter, which again is on the inside, composed of your horns. The ventral nerve root is for motor fibers, and your dorsal nerve root is for sensory axons. And then you've got your white matter, which is columns, okay? Those are the subdivisions for those. And then your spinal nerve plexes that I brought up before, those are at those enlargements, your cervical and lumbar enlargements. The purpose of those nerve plexes, which is your cervical and brachial plexi right here at your cervical enlargement, and then your lumbar and sacral plexes that are here at your lumbar enlargement. The purpose of those is because they organize nerve signals to kind of protect from any injury, and they do that by kind of like, a feedback system. They sort and recombine those nerve fibers from different spinal nerves. Big one we know that we're going to learn in clinic, and a lot of pre-meds will learn, it's called your brachial plexus. A lot of pre-meds will actually have to memorize and draw the chords of the brachial plexes. And that's up here. That's your brachial plexus, like this whole chord right here, which, gosh, is hard to memorize, I'll tell you that. But that's kind of the point of that, okay? And it looks like, yeah, these projections right here. See that stuff? Cool. So that is all the background information I have for you. So what we're going to do today, or this week, you have a virtual lab. Exercises 1, 2, 4, and 5. You're just kind of like following along and answering those. You can use my notes, obviously, that really help. And then exercise 3, which I have a screenshot of it below, it says you're going to watch this video. You click on the link and it takes you to the sheep brain dissection, which is a YouTube video. And from that, you're going to refer to the background information from that video, and you're going to label the diagrams with the appropriate structures, okay? Because in class, you would be able to dissect the sheep brain, but since it's virtual, you guys are going to do a virtual watch the video, okay? And that's pretty much it. As always, if you need anything, I am just one email away, and stay curious.